Okay, so does anyone have any questions for Pete then?
It doesn't look like it, I think.
Everyone's just being shy. No questions? How do you think the findings of there being like all these different regional groups of organisms, how that should feed into like the tourism industry, that they're not transferring stuff from place to place around Antarctica, or is that?
I think, that's been part of the emphasis for this sort of research. I wouldn't, whenever I talk about it, I don't really separate the tourism industry from the national operators. At the first level, it is simply awareness. If we know things are different, it's a lot easier to sell or get acceptance for the ideas that we need some sort of biosecurity when moving into new areas. To be entirely fair to the tourist industry, biosecurity seems to be more plainly applied in many tour industry situations, such as landings from ships or from airplanes. Boot cleaning is pretty regular, gear cleaning is pretty regular, and to be honest, the problem is more with some of the national operators than the tourist industry. But the awareness factor is the starting point. It's only 2 years ago or 3 years now coming up that Alex's paper was published. And we need to get that information out there widely in the public domain. It very plainly informs the sort of SCAR debate on conservation strategies and the recommendations that come from that in terms of moving between sites. We already have scientific recommendations for gear cleaning for moving between sites. It needs to be more widely applied.
Okay, thank you. Astrid, did you have a question?
I have a similar question. Hello, about tourism and biosecurity, and also the political implications of this research, because that would mean that there are higher conservation issues at stake than probably is realized by the purely political interests that have been maintained around Antarctica up to now. How do you look at this?
I very much agree. I mean, it is a far more important issue than had been realised until recently. Now, to be fair, I mean, there is a thing called the Antarctic Conservation Strategy being developed. Steve Chown in Monash in Australia is fronting that development on behalf of SCAR, but the development is to advise the Antarctic Treaty System, which is that international political body, if you like, that is responsible for the governance. Now, at one level, scientists can only make their results public and then make the governing bodies aware of it. Now, in the Antarctic, we're probably in a better position than in most of the world. There are actually quite a few. There are a small number of steps between the science community and the governance community.
Yes.
And SCAR, for instance, plus some various of the national operators and some of the NGOs are very keen that conservation of the Antarctic is a high priority. It's a very big responsibility under the Antarctic Treaty. So the mechanisms are in place, and I say the conservation strategy is very actively being developed within SCAR at the moment. There is a means to speak to all of those political influential people, if you like, in the Antarctic Treaty System. It doesn't mean they'll do what we want, but there seems to me there's more chance of that, and there's more chance of getting that evidence base in front of them within the current treaty system than there would be, for instance, if you were looking at all of Europe or all of Africa or something of that sort.
Okay, so that would be an advantage that the Antarctic has compared with the North Pole and surrounding areas.
Yeah, I think that's fair. Although the Antarctic system obviously has heavy political influence and all the historical factors and all the rest of it, it doesn't have the same national interests as you get in the higher latitudes of the Arctic. It's much harder to see a parallel system being developed in the Arctic than we could develop in the Antarctic. Yeah.
Thank you. Yes.
Okay. We've got some questions here that have come up in the question box. So there's a question here from Francisco Lara, who asks, why do you think that the mosses have such a low endemic rate?
It's a very good question, and there's not a straight answer. The basic reason seems to be that mosses actually evolve very slowly. So if you're not a molecular biologist and you look at molecular studies like this, it's very tempting to think everything mutates at the same rate. And therefore, if you've got a certain amount of sequence divergence, that represents the same time for all organisms, whether it's a bacteria or an insect or a moss or a human. That's actually not the case, and molecular biology doesn't assume that either, but an awful lot of people do assume it when they hear about molecular biology. Mosses, in the Antarctic certainly, mosses generally don't reproduce sexually. They reproduce asexually, so they're clones of each other for a start. One of the main sources of mutations in most organisms is changes that happen during sexual reproduction. So from a starting point, you might expect mosses to evolve quite slowly, simply because they rely on asexual reproduction in more extreme environments. The other reason is simply that mosses develop slowly, they live a long time, their generation time is potentially very long, and it does appear that their DNA mutates more slowly than flowering plants. So I put up an age estimate of 500,000 to 4 million years for the presence of that bryum species in the Antarctic. That estimate, the paper that we refer to there, does point this out. That estimate is based on a mutation rate from higher plants. That is because we don't have a mutation rate for that gene for mosses. It simply doesn't exist, and no one has done that measurement as yet. But the expectation is the mutation rate for a moss in a gene of that sort will be a half or less the higher plant rate. So that 500,000 to 4 million years is very conservative. It's likely to be double that or even more in reality. Now, that's actually a practical reality in many molecular studies. Although you do your best, or the scientists doing the study will do their best to give an estimate of age, we don't have mutation rates for every gene, particularly for non-standard organisms, but the non-lab rat organisms. So we have to infer, and that was an example of what was done in that study.